Hi Doctor, my name is Ellynwongzhuangee. For this assessment, I was required to select an open source software application and reverse engineer it conceptually. The software I selected is ImageJ, and I will walk through my full analysis. My presentation is structured into three parts. Part A covers foundational knowledge, what software engineering is, what ImageJ does, and the difference between requirement types. Part B covers system analysis, the functional and non-functional requirements, the architecture, and testing strategies. Part C covers software modeling, the use case diagram, use case specification, and activity diagram. Software engineering is not simply about writing code. It is a disciplined, systematic approach to designing, developing, testing, deploying, and maintaining software systems. The key word is here systematic. It means every step is planned, documented, and follow a process. This is what separates professional software development from informal programming. The framework that structures this process is called the Software Development Lifecycle, or SDLC. Looking at the table on this slide, there are 7 phases, and I want to explain why each one matters, and more importantly, why skipping any of them is dangerous. Starting with planning, this is where you define what you are building and whether it is feasible. Without it, a team has no clear goals and wastes time and money building the wrong thing. Then comes requirement analysis. This is where you document exactly what the system must do and how well it must perform. Skipping this is the most common cause of software failure because developers end up building a system that does not match what users actually need. System design translates those requirements into a technical blueprint, the architecture, data structures, and user interface. Without a design, the code that gets written has no structure and becomes impossible to maintain. Implementation is the coding phase. Testing verify correctness, deployment release the product, and maintenance keep it alive after release. The critical insight here is each phase is a quality gap. For example, if ImageJ developers had skipped testing, the measurement functions could silently produce incorrect values. A researcher measuring cell size in an experiment would get wrong numbers, publish incorrect results, and the entire study would be invalidated. This is the real-world consequence of skipping a phase in a scientific software context. Now, let me introduce ImageJ. ImageJ is a free, open-source image processing and analysis program developed by the National Institute of Health in the United States. It is written in Java, which means it runs on any operating system, Windows, Mac OS, and Linux without modification. It supports a wide range of scientific image formats, including TIFF, PNG, JPEG, BMP, GIF, and DICOM. What makes ImageJ particularly powerful is its extensibility. Users can install plugins and macros to add specialized analysis capabilities, making it adaptable for everything from basic brightness adjustments to complex fluorescence measurement and particles counting. The primary users are bioscience researchers, medical imaging professionals, and students in fields like science biology, neuroscience, biology, and physics. ImageJ solves 3 critical problems for its users. First, it replaces expensive commercial tools. Many labs cannot afford proprietary image analysis software, and ImageJ provides equivalent functionality for free. Second, it enables precise, repeatable measurements, things like area, pixel intensity, and length that simply cannot be done reliably by human eyes. Third, its plug-in system allows highly specialized analysis without programming. A biologist can perform fluorescence identification by installing a plugin without writing a single line of code. I personally download and explore ImageJ. The screenshot on this slide shows the main toolbar's interface, confirming that the software was successfully installed and launched on my machine. Before I list the specific requirements of ImageJ, I want to make sure the distinction between functional and non-functional requirements is very clear, because this is a conceptually important distinction in software engineering. A functional requirement defines a specific action or behavior the system must be able to perform. It answers the question, what does the system do? For ImageJ, an example is, the system shall allow the user to open an image file in TIFF, PNG, or JPEG format. This is functional because it describes a concrete capability opening a file. A non-functional requirement defines a quality constraint on how well the system performs those functions. It answers the question, how well must it do it? It does not describe a new feature. For ImageJ, an example is, The system shall open a 100 megabyte TIFF image within 5 seconds on a standard workstation. The key difference I want to emphasize is this. You can have a system that satisfies all its functional requirements but still fail completely. If ImageJ can open files but takes 10 minutes to load a large microscopic stat, researchers will abandon it. I will go through the specific functional and non-functional requirements I identify for ImageJ through my hand-on exploration. For functional requirements, I identify 4 core ones. Functional requirement 1 is about opening image files. The system must support multiple formats, including TIFF, PNG, JPEG, BMP, GIF, and DICOM, display with zoom and scroll capability. This was directly observed when I opened a JPEG microscopic image of a ficus leaf in ImageJ. Functional requirement 2 covers measurement. The system must allow the user to select a region of interest using tools like the straight line, rectangle, or ellipse and perform quantitative measurement including area, mean gray value, and perimeter. I confirm this by drawing a straight line on the image and triggering analyze to measure. Functional requirement 3 covers image processing operation, brightness and contrast adjustment, Gaussian blur, sharpening, and color separation, all with undo capability. I confirm this by opening the brightness and contrast panel and adjusting the minimum and maximum slider. Functional requirement 4 cover plugin management. The system must allow users to install and run third-party plugin without modifying the core application. This is confirmed by the presence of the plugin menu in the interface. For non-functional requirements, I identify 2 critical ones. Non-functional requirement 1 is performance. The system must open files up to 500 megabytes within 10 seconds. This is justified by the ISO IEC25010 performance efficiency attribute. Scientific microscopic stats are frequently very large and slow to breaks the entire research workflow. Non-functional requirement 2 is portability. The system must run on all 3 major operating systems with Java installed. This is justified by ISO IEC25010 portability attribute. ImageJ global user base span institutions with different OS environments and excluding any platform would exclude a large portion of the scientific community. For the architecture analysis, I identified that ImageJ used 2 complementary architectural patterns working together. The first is repository architecture. In this pattern, all subsystems interact through a shared central data store rather than communicating directly with each other. In ImageJ, the open image itself acts as this central repository. Every single tool, whether it is the brightness adjuster, the ROI measurement tools, or the particle counter, reads from and writes to the same image data structure in memory. When you apply a filter, the filter tools does not call the measurement tools directly. It simply modifies the shared image buffer, and every other component that reads from that buffer automatically reflect the change. This is why ImageJ tools are loosely coupled. They share data, not direct method calls. The second is microkernel or plugin-based architecture. This pattern separates a minimal stable code from optional extended functionality loaded as plugin. ImageJ code handles only basic image display and file input or output. Everything specialized. Fluorescence, quantification, 3D reconstruction, cell tracking is packaged as a separate plugin module. This is why the global scientific community can contribute new analysis capabilities to ImageJ without ever modifying or destabilizing the core application. The plugin menu in the interface is the clearest visible evidence of this. For testing strategies, I identify unit testing as critical for ImageJ because the measurement function must be mathematically exact. If the area calculation function has even a small rounding error, a researcher measuring cell size in square micrometers would get incorrect values, and their entire scientific study could be invalidated. Unit testing isolate and verify each calculation function independently before integration. I also identify usability testing because ImageJ users are scientists, not software developers. The developers would have tested whether a new user can open an image and take a measurement within minutes without reading a manual. This mapped directly to the usability qualities attribute in ISO IEC25010 covering learnability, operability, and user error protection. For the use case diagram, I identify 1 actor and 6 use cases based on my direct exploration of the software. The actor is the researcher. This is the single human actor for ImageJ. There is no server, no administrator, and no secondary actor because ImageJ is completely standalone desktop application. The researcher represents any scientist or student using the software. Let me walk through each use case and define the relationship. For use case 1, open image file is the mandatory entry point. Every other use case depends on having an image open first, so this is the foundation of the entire diagram. Use case 2, adjust brightness and contrast has an extend relationship with use case 1. I choose extend and not include deliberately because adjusting brightness is optional. A researcher may open an image and go directly to measuring without ever touching the brightness and contrast panel. The extend relationship correctly models this optional behavior that is only triggered under certain conditions. For use case 3, measure image is the core analysis function, and it has an include relationship with use case 6. I chose include here because selecting a measurement tool is mandatory. You cannot trigger analysis to measure without first having a tool selected and a line drawn. The system physically requires it. This is directly confirmed by my hand-on workflow where I had to select the straight line tool before I could draw anything. Use case 4, apply image filters also extend. For use case 1, it is optional and only occurs after an image is loaded. For use case 5, save export image is standalone. It cannot be triggered after any modification or measurement. For use case 6, select measurement tools is the subset included by use case 3. During my observed workflow, the straight line tools was specifically used. I select measure image for the detailed use case specification because it is the most central scientific function of ImageJ and it was directly observed during my hands-on exploration. Looking at the table, the precondition established that ImageJ must be running, an image must already be open, and measurement parameters must have been configured. The brightness and contrast adjustment is listed as optional, consistent with the extend relationship in the diagram. The main flow has 5 steps. The researcher selects the straight line tools, draw a line on the image. The system highlights it with a yellow overlay. The researcher presses Ctrl+M and the system populates the result table. The actual observed value from my exploration were error equals to 261, mean gray value equal to 197.259, then equal to 260, and angle equal to negative 90. These are real value I obtained from the focus leaf microscopy image, which validates the entire workflow. The alternate workflow are important to mention because they show I understand the system edge cases. Alternative 1 covers the case where no line is drawn. The system measures the entire canvas. And alternative 3 is particularly interesting. It confirms the ImageJ can measure an RGB color image directly without requiring grayscale conversion, which I verified because my test image was a 23 MB RGB file. The activity diagram models the complete step-by-step workflow for the measure image use case. It has two swim lanes. The left lane belongs to the researcher and the right lane belongs to the ImageJ system. The workflow starts at the blue black circle, the start node. The researcher opens an image file via file to open and the system automatically displays it in a new image window. Next, the researcher opens the brightness and contrast panel and adjusts the slider. The system immediately applies the enhancement. Then the researcher selects the straight line tools from the toolbars and draws a line on the image. The system responds by highlighting the line with a yellow overlay. Here we reach the first decision node. The condition is, was a valid line drawn? If no, the flow loops back to the draw step. This loop correctly models the real behavior of ImageJ. If you click without dragging, nothing gets measured. If yes, the flow continues. The researcher triggers analyze to measure. The system calculates area, means gray value, length, and angle. Then we populate the result table with a new row. We then reach the second decision node, measure another region. If yes, the flows loop back to the select tool step, allowing the researcher to measure multiple regions without restarting. If no, the researcher optionally export the result table to a CSV file. The workflow ends at the end node. That's all from me. Thank you for watching.